Hey everyone, so today we're going to discuss X inactivation and X-linked inheritance, as well as show an example branch diagram depicting different traits in cats. The line hypothesis is a hypothesis that explains gene dosage imbalance between males and females. As you all know, this X and Y represents a male chromosome, but this, two X's, represents a female chromosome. Females have twice as many copies of all the genes on the X chromosome, and they generate twice as much protein because of that. But during the inactivation process, instead of having double gene expression, they will only have one that's allowed to be expressed. Once activated, all progeny genes have the same X inactivated, and then inactivated X is called the bar body. The bar bodies are inactivated, tightly compacted chromosomes that have very little protein gene expression, and they regulate the amount of X-linked gene product being transcribed. So now we will transition into the process of tortoiseshell phenotypes in cats. So, as you all know, two big B's would represent the dominant trait color, black, in cats, while two little b's would represent yellow, which is the recessive, not as common color. In fur, Big H would represent the dominant long hair trait, while two little h's would represent short hair. Together, a big H and a little h, as well as a big B and a little b, would represent the tortoise shell phenotype. As you can see, this is the combination of the phenotypes that different parents of one dominant black long-haired cat and one recessive short-haired yellow cat would have as children. Their color would represent all tortoiseshell. They all would be represented by big B, little b. However, the hair would be represented with a 50% chance of having long, 50% having short. This can be depicted in the branch diagram. As you can see, half long and half short would be multiplied by one tortoiseshell. This would give us a 1/2 to 1/2, 1-to-1 phenotypic ratio. A clone tortoiseshell would not be the same as the mother, because the expression of certain genes in clone animals can have as much variation as within naturally bred animals. Due to the external and environmental factors, some genes may be inactivated and others may stay activated in some clusters of the skin cells. But a clone would never be identical to the mother.